Good morning, everybody. Thank you for being here. And as we mentioned before, Dr. Lucio Lombardo will start the presentation talking about intensive breeding as it is being carried out in his team. And then Mr. Bossio with his part of the research. Lucio, the floor is yours. My thanks to Aapresid and for the people that are joining us. It's a great pleasure to be here. Let's see how I can... Sorry about that. The title of the presentation is Intensive Breeding Applications for the New Climate Scenarios. And this has to do with the time in the breeding. I'm sorry, if you can help me out with the laser pointer to see if I can move the presentation on. Okay. As I was saying, currently time is of the essence because why? Because we are undergoing the climate change, and that will result in changing the rules of the game. And there are a couple of things that I would like to mention. For example, increase of disease incidence caused by fungi, change in insect population, in weed population, changes in drought schemes that will lead to drought, and soil composition and salinity that has to do with the rainfall. And that will have an impact in the agro-bio-industrial change. So we need to find different strategies to address climate change. And this includes mitigation, reducing the carbon footprint, and adaptation, that is to say, find ways to minimize the impact. And when we talk about genetic breeding, getting adaptation is to have more intensive breeding that are focused on climate resilient and related traits. And the question is, how do we do that? Well, and here we have the breeder equation. Genetic variation is directly proportional to genetic variation, the selection accuracy and the duration of the breeding cycle. So we are still in primary school. We need to increase the numerator and reduce the denominator. There are different techniques for this. The three in green, for example, that reduce the breeding, or the double haploids and the speed breeding, fast phenotypical selection, or assisted with molecular markers, mutagenesis, transgenesis, and genic addition that increases the genetic variation. So let's go very quickly and see the different techniques. So we need to move the germoplasm from one hemisphere to other for backstations. We can also move the germplasm to warmer areas, and this would imply, for example, well, I like to clarify that actually this technique of moving the germoplasm is not to reduce the breeding time, but not to lose time. If we want to reduce the breeding time, we need to use the speed breeding, for example, which is an indoor crop technique for the stabilization of traits. And this is an example, for wheat. And this varies depending on the crop. For wheat, the plant is subject to vernalization, then extended light period, and we have a crop a cycle of 3 months. So we reduce it by 50% from 6 to 3 months. And this technique is a very powerful technique because for certain germ plants that do not require cold, we can reduce it to 2 months. So we can have 6 generations in 1 year. The speed breeding also can be phenotyped, and we turn it into a fast phenotype. For example, we see the plant height and the different disease. Another technique that I mentioned before is molecular marker assisted selection. And in this case, we don't use the phenotype. We will define whether the plant has a desirable agronomical quality. And we can do this, for example, without presence of the pathogen, because I can choose a gene that is resistant to disease. We can select recessive traits, and the most important thing is that we do it regardless of the climate. That is to say, we don't pay attention to the environment. So this assisted selection example is applied to different variants, for example, recurrent variation, or other types, or the genomic selection. But the concept is always the same, that is to say, to see if there is a desirable gene. And then mutagenesis, because the next presentation is going to talk about addition to genesis. And mutagenesis implies exposing the seed to a chemical and physical agent and create random mutations. And at the end of the day, this will result in will appear in the gametes, and we will be able to select. In Marco Juarez, for example, we apply bottleneck effects, and we use the plants that survive this treatment. And the importance about this technique is that we select the seed, that is to say, elite genes. I like to explain this. For example, I have a problem with my TV set. The TV set is broken. If I know about electronics, well, I can change myself the components. Or if I give just 2 or 3 knocks to the TV set. Mutagenesis is like that. When we know what we need to touch, we go to transgenesis. And mutagenesis is an old tool, but we are still using it. So these are some of the future problems. For example, the different insects, weeds, fungi, the different tools that we have to address this. For example, for fungi resistance, clearly literature indicates regarding climate change, and that we know is fusarium and the 3 rusts, and we have molecular markers. We can select markers for resistance, and in Marco Juarez we use 2 strategies. For example, we use molecular markers for back crossing, and then in speed breeding of segregating populations, that is to say F1 or F2, we select the materials that have these markers because we have other markers as well, for example, and the other thing that I mentioned has to do with resistance to insects. We can see the problem of the different bugs and hemipterous and being replaced by phytophagus, for example, the corn bug, and we are using 2 strategies, mutant populations, and we do this with Dr. Vicentin from INTA Paraná. We have identified a good mutant, and then another study that is being performed with soybean genome, for example, resistance in exotic soybean. And we have developed molecular markers that are being used in speed breeding. And the other thing that I mentioned that are related are salinity. As you can see in the Orione Basin close to Córdoba, there are a lot of salt ponds, and we have selected mutant resistant, and we are using these with the INTA breeding program. And we are also doing this in San Juan. We have selected for plants that are resistant to droughts, and then herbicides or weeds. And in this sense, we are talking about herbicide resistance. And here we can see the different effects. And what we see here is the effect of the resistance. But this material is also being used as breeding material. But I mentioned about the breeding time and how important it is to act quickly, particularly in the face of climate change. And this is a genotype method that we carried out with CAD, the germination plate. You can develop, you can download the file. This is a germinator, does not affect the root, does not create fungi. And as you can see in the left, this is a bioassay where we use saline solutions, herbicide solutions, or PEG solutions, and we can see the effect. A material that is resistant to a herbicide and a susceptible material. Otherwise, we need to carry out other type of tests. We can put them in a cardboard with an air reference, like the red box, and with a photograph and a pixel counter, we can detect the biomass. We have the dose response rates, and we can determine whether the plant has the resistance or not. The importance about this is the time. It takes only 8 days. We can recover the plant, we can place it or transplant it because the roots remain intact, and we can continue the breeding process. Going back to the topic of this presentation, this is a wheat breeding program. I come from the wheat breeds, basically, and the minimum duration is 12 years. We have variability, generation, self-fecundation, stabilization of traits, multiplication, assessment. There are initial assessment, preliminary, and more advanced assessments. The problem with this type of schemes is that it's tied to the climate. If we have some weather problems, if I want to, for example, assess resistance to a disease and I don't have the disease, well, probably I will have to sow again and spend 1 year more waiting for the results. So we will create these, or we will do this in a more intensively manner. For example, in 7 years to generate the germoplasm, we can replace this with speed breeding and contestation, and this would take us 2 years. And we incorporate molecular markers or genotype fast selection method, and we can reduce 2 years. And if we increase the number of sites of the assessments, we can reduce it 1 year more. From 12 years, we move to 4 years, 4.5, 5.5 years, let's say 6 years. But we reduce the environmental risk, and this is very important because climate disasters make it difficult to follow the traditional pattern or scheme. So we need to get rid or to not to think about the weather. So to summarize, so the breeding results will result in climate independence. Yeah, the plant has to be in the field, of course, but we have to reduce contact with weather at certain times of the breeding. Then incorporate environmental variations to the analysis. We are talking about environmental typing, interdisciplinary. So we need to incorporate different experts from different topics. Well, agricultural scientists, biotechnologists. Well, here we have three: statisticians and climate experts and IT experts, because AI is part of the equation. Well, we are going to work in climate resilience, and there are different alternatives to address climate resilience. We can use old material that agronomically is not enough, but can provide some genetics that is useful. We can use exotic material, for example, the example of the soybean, and because we can consider past problems and use them for the current problems, and mutagenesis, transgenesis, and gene edition. And the proposal is that each breeding program should focus on specific targets and microenvironments. It's impossible to have one single plant for all Argentina. We will have to address these on a problem-by-problem basis in order to consider the yields. And this is a prediction of artificial intelligence. We ask, make a scenario for improving the wheat scenario. And this is, it looks like an astronaut, but at INTA Marco Juarez, we are sure that this is the right path. And we recently created an interaction PPP platform where we provide different type of services to those that are interested to use these type of services. We are carrying out training sessions. We develop technology like the germination plate and developing germoplasm using this technology. So this is my contact information, and we will be happy to answer any questions. Thank you.
Muchas gracias, Lucio.
Thank you, Lucio, for your presentation, for introducing the platform. This is very interesting. And now we give the floor to Ezequiel, who will talk about gene editing and transgenesis. Good morning, everybody. Thank you for inviting me to participate in this panel and share with you what we are doing at INTA. Well, what Lucio said or told you is the foundation for my talk. I'm going to tell you how we use biotechnology tools to cooperate with the breeding programs. The goal of my presentation, these are the themes I'm going to talk about: molecular biology, modern biology, the scenario in Argentina, and elsewhere of biotechnology. And now, finally, we'll talk about gene editing, talk about specific variability, and finally tell you what we are doing at INTA, at the Genetics Institute. FAO says that there are technology uses everywhere. Technology was used. Biotechnology was used all over mankind's history to make bread, to make beer. Today I'm going to talk about biotechnology tools that are used for the breeding programs. This is a list of, well, can't see the pointer, but it's a list of biotechnology tools that have been used in breeding programs for a long time now. Hybrids, molecular markers. The final group is the omics and their contributions for genotype selection. I'm going to focus on modern biotechnology. where on one hand we have conventional breeding and assisted selection using molecular markers, and on the other hand, modern biotechnology, which uses gene engineering, and this is related to the use of GMOs in agriculture. Biotechnology is the technology that had the fastest adoption in agriculture in the last 20 years. The presence of increased, the growing presence of GMOs has been really fast. You can see the word figures on the screen, but there is a limited number of crops. We can see that there are several events worldwide that have been authorized in several crops, but mainly in 5 crops: cotton wool, soybean, corn, alfalfa, and rapeseed. And what about Argentina? Since the first event was launched in Argentina, 77 events were authorized, basically in soybean, corn, and cotton, and fewer events in potato, alfalfa, wheat, and another crop. And there are very few crops that are using modern technology for breeding. And I'd like to talk about traits now. You can see that there are very few traits that have been dealt with: herbicides and pest tolerance, together with, well, joined or individually in the 3 crops. Going back to what Lucio was saying, here on the screen we can see the various tools used to introduce variability. I'm going to talk about modern technologies now, working with the genomes, and the focus of my presentation is gene editing. What do we mean by gene editing? It's a biological process that enables us to introduce specific interventions in the genomes, and it can be used in many different ways. Every day we find new ways of using gene editing. We use it to do away with segments or to substitute nucleotides to create new variabilities in a specific fashion. It's important to stress that gene editing uses several tools. The earliest ones were the zinc fingers or talents, and later on we had CRISPRs related to Cas9. These are the best known ones, but there are other tools beyond CRISPR. Where does CRISPR come from? It's a biological system which we use. It's related to prokaryotes. It's a tool that has the bacteria to create an immune system that stops virus replication by using some segments of the phages. And once a phage has a similar information, that machinery cuts the DNA and destroys the virus genome. So we use this approach to bring about specific changes in the genome in specific sites. This is gene editing used as a tool. We create these segments of ARN that tell us where we have to carry out the intervention related to the Cas, an enzyme which cuts the DNA, and after this we go to a specific site in the genome and break it. And the cell machinery of all cells, in this case plant cells, repair the DNA DNA that we have broken. Usually the DNA breaks, and there are many enzymes that are continuously repairing the DNA when it breaks. But in this case, we broke it in a given place, and the machinery of the cell will repair this DNA, and through this process, either everything goes back to baseline or repairs this in a different way. In this latest approach, new segments are created and the gene does not work or operate. It used to operate before the intervention. How do we carry out gene editing? Well, I'd like to clarify this. If you have read that carrying out or doing gene editing is easy, well, don't believe this. Based on our expertise, we may say that maybe it's simpler than doing transgenesis, but it's not simple at all. We have all these stages from time zero when we plan a new research project until we get the plans, go through all these stages. When we talk about bio IT, we define the machinery so that the intervention is carried out on a specific site in the genome. We already have the places where the DNA is going to be broken, and we have to define the guide or guidelines. So we have to find information about the gene on which we want to act. We have to find out the sequence on which we want to work, and with that information we have to define the guidelines. Once we have the guidelines or the guides, we go through a genetic engineering, so we design the vectors that take what we want to take to the core of the cell. Here we already have the biological information that codes the enzyme for the CAS and some other elements that are typical of the DNA transfer system and plant recovery system. So through the vectors, we add the guides through the vectors, the ones we had already synthesized. This is done through cloning, a cloning process. And this is the vector that carries all the biological information required to go to one point in the genome and carry out an intervention. Next step is where we have the vector, which is ready to be used. We have to take it to the core of the cells, the plant cells in this case, so that the editing machinery does what we expect it to do. So first of all, we have to develop an in vitro culture system for tissues, and after that we have to take this editing device to the core of the cell. And in grasses, which is our main area of work, we use certain cells in the culture that come from some embryos. These cells are cultured in vitro and they may give rise to a whole plant. What is interesting here is that we may work on cultivars that have already been improved. In the past, we were forced to work with very old crops. Currently, we are able to work with lineages, new cultivars, or specific lines that are improved breeding foundations, so to say. So we take cells coming from other genetic groups, and we may use two methods: bombing with gold microparticles. In that system, we put these vectors on very tiny gold particles and we shoot them towards the plant cell, so that the gold particles go to the core of the cell, and the vector is therefore inside the core, the nucleus, and the machinery starts working. The second method is the biological one. Depending on the crop, we use any of these two methods. These are some images that show our work. This is the lab of the Genetics Institute. Here we have the technicians working in isolating cells. My colleagues are in the room right now, here. Based on these basic cells that we isolate, we cover the complete or the whole plant through an in vitro process. And in this process, at some point in time, we use a particle bombing approach, and so the plants have in their genome their editing machinery, and if we are lucky, the editing has taken place. So what do we expect to happen? Well, we want to get plants which in some part of the genome have the editing machinery, and in another section where we wanted it to happen, we have the gene editing. In red, we have a place of insertion of the vector carrying the biological information for the editing machinery. We cannot predict where the vector is going to land, but we can predict where the gene editing will take place, which is actually what we are interested in. That is the green line on the right. So after that, we have to divide the plants in two groups, those that are transgenic and those that are not. So we use molecular selection for this. Through simple PCR, we identify the plants that have the editing vectors, and through PCR, we look at the place where we want the gene editing to take place. After that, we do the sequencing of the areas of interest where we hope the gene editing has taken place. We use the Oxford nanopore system, and this is what we get. This is an example of a wild-type plant, a genetic element where no gene editing has taken place yet. The gaps you see there are the mistakes or the errors of the sequencing. Here we can see the edited plants. These are the gaps we create through gene editing. These are elements where some DNA segments are missing, so the gene which we are targeting stops operating. These are wheat plants, bread wheat plants, and the editing was done in such a way that each allele was intervened and repaired as well as possible. So we have 6 modified alleles for each gene and 6 different interventions of the editing machinery. This is another example with wheat, and this is barley. Here the sequencing looks simpler because it's diploid and not hexaploid. We can see a line there, what you have at the top in red. This is the editing line with just one guide. One base was deleted, so the framework was moved, so this gene stops operating. And the deletion can be found in 50% of the readings. So one of the alleles was broken and the other one wasn't. Why are we interested in this, and why do I talk about making a distinction between transgenic plants and those that have just been edited? In Argentina, there is a regulatory framework that governs the activity related to GMOs. So, and special licenses are required to be able to release this into the ecosystem. And we need special licenses to sell this product later on. Starting in 2017 and 2019, the two amendments, we have regulation in Argentina and elsewhere that makes a distinction between GMOs and NBT developments or other ways of breeding improvements. This is based on the Cartagena Protocol. GMOs are all individuals that have a new combination of genetic material. So if to these plants I was talking about, if we remove what we inserted, that is the vector, the editing vector, and we have plants where the only change is the editing we did, we would have individuals that don't have a new combination of genetic material. So these individuals are not GMOs and would not be under this regulatory framework. So once we complete the gene editing, we want to do away with the vectors. We do this with a simple segregation, we plant the plants and we remove those that have the vectors and we just keep the ones that have a genetic foundation where the editing has been done. We plant them in a biosafety greenhouse and we use something similar to what the previous speaker described, Lucio. We speed up generations, so we speed up generations because by using immature embryos. This is data for intermediate cycle crops, spring crops. So we have 4.5 cycles a year. This speeds up the whole process of cleaning the genetic foundation and getting plants which are not GMOs but which have been subject to editing. We do a molecular follow-up to just keep the plants we are interested in. And in closing, I'd like to tell you what we are doing nowadays. The idea is to have new technologies, and we want to have, as we have an agile regulatory frameworks, we were able to exchange ideas and develop new research lines and areas which were launched 4 or 5 years ago. We are working on gene editing in wheat and barley. Some of the results I've shared come from these lines, and early this year we started working with rice, and in all cases with the breeding programs, working together with those people in charge of breeding programs in wheat and barley. We started doing this in rice a short time ago, so we don't have results to share yet. By way of conclusion, well, biotechnology has made a significant contribution to crops productivity, but we are still working on a very limited number of crops. The number of traits analyzed is limited to. This is technology which is not very interesting for minor crops. Beyond the R&D costs, we have the issue with the regulatory frameworks. Not all crops are so profitable that it's worthwhile working in these areas. Gene editing and other regulations offer a new approach so that we can use biotechnology for crops which haven't been dealt with yet. So with the new technologies, we are able to work using now we are able to work in other crops and new traits. Finally, this is our team. Almost all the members of the team are in this room now. This is the group working in wheat, barley, rice, and cotton wool, we have the people working in the warehouse, sorry, in the greenhouse and in the lab. Thank you very much, and well, I hope we have time for questions. Thank you, Ezequiel. There is a lot to be said about biotechnology, of course, and what INTA is doing. This is a very interesting subject matter. We don't have too much time. I don't know what happens with questions. Is there a QR for people to send questions?
Mientras tanto, yo tengo algunas preguntas.
In the meantime, I have some questions that I would like to ask the panel members, taking advantage that Lucio and Ezequiel are part of the panel. Lucio, with regard to what you were saying, where can we focus the breeding in the short term, in the speed breeding, where we need to focus the speed, let's say? As I mentioned in my presentation, getting climate resilience, we are talking about thermal stress and water stress. But personally, what I think the improvement should be based on the breeding background, because the breeder always saw the plant is okay, looks good, always from the soil upwards. But we didn't pay attention to the root. I think that we need to focus also on the roots. And when we look into the future, the roots play an important role. Thank you, Lucio. And I have a question also for Ezequiel. Then we will see if we have questions from the audience. But Ezequiel, any gene can be edited in any part of the genome? Yes, any plant can be edited. Initially, yes, but what we need is that has a good in vitro response. The editing requires good performance in vitro. Many years ago, there were many breeders and crops that were impossible to do them in vitro. But today, many technologies are more modern and sophisticated, and we can regenerate plants from any crop. So the answer is yes. And with regard to the genes, I didn't mention how we design the guidelines, but if we need specific traits to put the guidelines in the editing place. But if we have the genome information, we can have the guidelines, and then we can intervene wherever we want. Selena, for example, has a question. How stable is the gene edit? Do we know how many generations? Yes, it is very stable as any other modification, like in mutagenesis, for example. Once we recover the plant and we make the sequence and see that there is an editing, that modification is inherited as in with any other allele. It's a stable modification. I don't know the number of generations exactly, but once the generation is stable and we sequence it, it becomes stable. Another question for Lucio. How is the mechanic for the resistance to one of the bugs? That is a very good question. We are investigating this. The soybean grain is a little bit smaller. So what we want is that the bug fillet does not touch it, because there is a change in size. But soybean has recently being included in the mutation population only 2 years ago, and we are now making the research and see how we can implement that in the breeding program. Now, another question for Ezequiel, because now we have to conclude the panel because we are waiting for the next panel. Thank you very much. You can contact the panel members at their stands. So the last question for Ezequiel is, today I understand that we are in the research stage in gene research in crops. When will be able to see commercially available crops with gene mutation? Yes, edited crops will be part of the market because laboratories are performing a lot of tests, and the advantage is that we work in already improved genetically material. We are already working in modern crops, so we get seeds from a breeding program, for example, with the last encrypted variation, and we develop the same genetic foundation, let's say, and the genetic foundation is already improved. That shortens the time frame, and in the near future we will see edited variations in the market. Thank you, Lucio. Thank you, Ezequiel, and thank you to the audience for attending this presentation.